So let me just, before I go into the system itself, I wanted to talk just a little bit about one of the benefits of using Biodata Catalyst, just from an overall big picture vision perspective. There is so much data available in a lot of different human studies, specifically focused on specific diseases, but also some overall cohorts who have very specific populations of interest to study. So just a couple that I've highlighted here, 1,000 genomes and a Parkinson's disease data sets are just one of many. The value of having this centrally located on a Biodata Catalyst environment means that all of us don't have to have all of the server space and the server farms to be able to get access to this data. So being able to have this data and house it, but yet selectively choose what data you'd like to look at is really a huge advantage of Biodata Catalyst. So just a couple things here, and I've shown you just some data that I have access to that we used as part of our study that Maggie just mentioned in terms of the HCHS SOL data. The access that you have in Biodata Catalyst, so even though the data are housed, many different data sets are housed on Biodata Catalyst, the access to those is still governed by your dbGaP access. So whatever you're entitled to based on your relationship as a PI or your working relationship with a PI, that is the data that you have access to. And so even though this huge data set of data is available to many different people, it is still governed by the rights and privileges. And so the data are very protected for the use in your studies, especially since most of this data is related to humans. And in particular, whole genome sequence data is just large, very large. And so trying to house that yourselves would be very difficult. So I'd like to take you into the system itself and show you, even though there's a few slides that will be available to folks, I'd like to walk through the system and then only head to those backup slides if we need to. Certainly would Q&A, okay, that's perhaps a later time. Oops. Make that go away from my screen. Okay, sorry about that. So I wanted to take you into a particular workspace that I have access to. This is where we did some of the polygenic risk score work that Maggie was discussing. So I am logged in here as myself. And so the data sets that I have access to are part of this logon permission structure. And then within this area, I have a particular workspace where we did the polygenic work. So what you'll see here are a couple of different tabs associated with this workspace, just general workspace information, where it's located on the cloud, etc. But one of the most important things are the data that I have access to. And one I will show you in particular is the extract SNPs. So as Maggie mentioned, we were doing work with a polygenic risk score. And so that polygenic risk score is made up of many different, very specific nucleotide positions across the genome. So we were interested in grabbing the values associated with that genetics for our individual participants in our study. So we've got data sets set up for those specific individuals in our broader SOL study. This particular data set that we've entitled Extract SNPs has basically a different set, a different row for parts of each chromosome. So this was how we organized our data for this particular study. Each of these rows represents a portion of a chromosome. As I mentioned, these are very, very large data sets. And so in some cases, dividing them up into different pieces is important just for sheer time factors associated with manipulating the data. So in this case, we're actually looking at output. We're grabbing all of the scores or all of the allele positions for those particular SNPs for our asthma study, and we're storing it in a file name. So we don't need to necessarily know anything about exactly where the data is. We can just reference it using this table. So you could think of this table as being kind of an index into the data that we're interested in looking at for our participants. So some important things associated with that data that we'll need as we're accessing it. But also I wanted to show you a couple of other things. So in the case of some of these fairly simple looking data sets, this is a list of the nucleotide positions that we'd like to extract, but we're accessing this bigger data set. Let me see some things on my screen here. We're accessing this much larger data set, which is actually the overall SOL data for that chromosome portion, but it is housed in a central location. So even though this data set, which we're using to extract a list of SNPs, is something that we have created ourselves, we're accessing something that's been created by the SOL study. And so we do have access to that because of our dbGaP privileges, but that's a single copy that is located and available for anyone else who might also have access. So we're using that as part of our analysis, but we don't have to separately house that data. So I also wanted to show you the workflows that we have used. This is just a series of different workflows that we've created to be able to manipulate the data that we were accessing from that data table. So let me look at one in particular, extracting those nucleotide positions. It starts with, so in Biodata Catalyst, powered by Terra, which is what we were using in our particular study, a workflow is called a WDL, WDL, workflow definition language. And in that workflow, we're using pretty much standard terminal level commands. If any of you have worked at the terminal level in other environments, it's fairly rudimentary in terms of its capabilities, not for the faint of heart, but fairly standard in terms of what you would find in this environment versus another. However, so lots of workflow definition to show which files we're pointing to that we will be using as part of executing this workflow. But down at the heart of this, WDL is actually a standard Plink command. So some of you who have worked with Plink in other environments will recognize some of the options that can be specified as part of a Plink command structure. So pointing to a particular file, using particular options, extracting from a range of SNPs that you're interested in pulling out based on those extraction files that we defined, and then defining where to place the output. So the key thing about this is that this is a generic script that can then be used on many different data sets. And so the input to that script is specified in a data table. So the data table that I was just showing you a few minutes ago is accessible from within this script environment. So I can select data and actually choose individual elements that I would like to work on. I usually go for something like chromosome 22 because it's the smallest. So that script, which was specifying inputs, one input was this VCF file that we've noted here. And an output of this is going to be this traw file. So regardless of which row I pick in my data table, that script will operate on that particular data set. And so by clicking OK, I have selected a few, that particular row to be executed. And the input values are specified as part of the script, but then also defined very specifically as part of the input tab within the WDL. And similarly, the outputs are defined in a similar way. Then to run that analysis, and I would say community hours here, of submitting our job and launching that in the background. So the nice thing about running a WDL and launching a job is that you can now go on to do other things. So what you see here is the job that I just started. I just submitted it. And so it will be in blue until that job finishes. And below that, you see a couple jobs that have completed successfully. I ran this one just trying to test for this particular meeting today. Let me move my people around. And so you can see that this job succeeded that I ran a couple days ago. So, and then there's lots more information associated with the job, outputs, et cetera, that are a result of that. And so that really is the set of steps that we used repeatedly when we were conducting the asthma study with the gut microbiome, by being able to, but the heart of the use of biodata catalysts in this particular case was to be able to get access to the whole genome sequence data in an efficient manner. So that's really my 2 cents on a bit of a dive down into the structure. And again, going back to the access to the system. It's all powered by the access and privileges that you've got as part of being a Biodata Catalyst member, and the access that you have in dbGaP as well.